Okay, so we seem to have got a reasonable number of people who've kind of trickled in. I guess some more will probably join as we go on. So for anyone who wasn't here at the start, welcome to this Theory of Living Systems webinar. And we're very lucky today to have Adelle Costa from the School of Mathematics at UNSW, who's going to talk to us about location and timing of regulation and control of insulin signaling pathway. Adelle works on kind of a wide range of interesting topics, including identification of ancient starch and the interaction of actin and tropomyosin. Yeah, I'm really excited to hear what she's got to say today. So yeah, if you want to take it away, Adelle.
Thank you very much. Yeah, so this is one of my main interests that has been for some time, and that is looking at the dynamics within a cellular signaling system, in particular the insulin signaling system. So I'm throwing up here just one of the diagrams that you can find on the web about the insulin signaling pathway. So in this system, you've got insulin, which is synthesized in the pancreas. And when you get glucose in your blood, say you've had a coffee like I've just had, then glucose in your blood is detected, and that causes insulin to be released, and it flows around through the blood to various cells in mammalian bodies like ours. And these are predominantly fat and muscle cells that respond. So the insulin binds to the insulin receptor, and it causes a whole cascade of really interesting events. Now, the thing is that there's a large number of components here, okay? And there's lots of lovely boxes and arrows. And indeed, if you look around, there are some that are repeated. So there's an AKT here and an AKT here, and there's an AKT down here. I'm specifically mentioning AKT because I'm going to talk a little bit more about it. The point is that it's complicated. And also, we only get to look from the outside in. In a living cell, there's limited things that we can do to actually look at the dynamics well in order to infer what is going on. So what this is supposed to illustrate is, A, it's complicated, and B, there are a lot of players that we know about, biochemical components, which we know are actually useful things within this signaling network. What we don't know precisely is how it all works. So whilst this might sort of indicate that we have some knowledge about this, actually it's relatively sparse. And we might know what the things look like or what the key players should be that are in our model, but we don't know what the interaction should be. And we also don't know precisely the order in which these things occur. So it's interesting then to say, well, you know, if we've got lots of measurements that can be done from the outside of the cell in, so this is the outside of the top, and you've got this membrane, which is a bag around the cell, and you're looking from the outside in, there's many unobservable features. And so we have to take a parsimonious approach when we look at modeling in these sorts of systems, because you might be able to take lots of measurements, but what you can observe and what you can't observe are not equally distributed. Okay, so I'm going to be particularly focusing on a couple of components. And the first one is AKT. Now, there's actually 3 isoforms of AKT, and I'm just illustrating AKT2 here because it's the particular form of this biochemical component which is important in metabolism. So this is one of the key critical nodes. It has a lot of crosstalk actions. So it's also known as protein kinase B, or PKB, which for a naive person coming into the field, lots of these components actually have multiple names and they're all the same thing. And so you can read lots of papers and say, oh, that's another interesting component I should put in my model, but it's actually just another name for the same thing. So a key trap for new players. So AKT plays a pivotal role in glucose uptake. It's also a key node for cell survival, proliferation, and it also does responds to numerous growth factors, And so it's part of that regulation system, not only for the response to glucose coming into the system, but also other key processes in the cell. So that implies that dysregulation of AKT will have a whole lot of on-flow effects to a variety of disorders. So you see changes in AKT dynamics and expression having ramifications on type 2 diabetes, on cardiovascular disease, and also in cancer. So it's a very important molecule that needs to be tightly regulated in healthy cells. So for me, I'm mostly interested in the metabolic pathway, so glucose transport. So it is a key stop node that stops us from being able to transport glucose in and out of the cell, which is done via another protein, glucose transporter protein 4. There's plenty of isoforms of that, but the insulin sensitive one is GLUT4. And it is effectively the tube through which the glucose can flow in and indeed out of the cell. Now, activation of this molecule AKT is both necessary and sufficient for that downstream GLUT4 translocation to the membrane where the glucose can then flow. So interestingly, it's been found that AKT acts as a very, very low threshold switch in controlling that translocation of the glucose transporter protein. And so actually an activation of a tiny fraction of AKT actually allows maximal translocation of glucose transporter proteins. So you can activate AKT via other mechanisms other than the insulin signal, and indeed that is sufficient to trigger the downstream response. So it's clearly an interesting molecule for lots and lots of different reasons. So we need to take a cleaner approach, take a Occam's razor approach, put in as much as is sufficient to explain the system and not more, and that way we might get some insight into what's happening in this system. So AKT is found in all sorts of different places in the cell. So it's synthesized inside the cell, that's in a leaflet down here of the nucleus, and it then forms an activation complex with another molecule, which is PDK1, and it's in the cytosol then, which is the inside of the cell. So the outside of the cell is up here in this blue part. You have the plasma membrane, which is the outside membrane of the cell, and you have a nucleus in the middle. So we have our insulin floating around. It binds to the insulin receptor. It comes in and creates a cascade of phosphorylation events which activate different components and provide a biochemical signal down to the system. In response to that signal, AKT is known to move from its inner position inside the cytosol of the cell, inside, to the underside of the plasma membrane. It is at this location that it gets phosphorylated. So it actually gets phosphorylated twice. That is, a phosphate group gets chemically added onto it. It is then in its activated form. Now, the activated AKT is found both on the inside of the plasma membrane, but also within the cell. So both the active and the inactive form of AKT are found in different locations within the cell. But we do know that the activation occurs only on that underside of the plasma membrane. Now, that activated AKT then signals downstream. So the metabolic effects, like transporters for glucose being moved around in the cell, happens, and that's one of the things I'm particularly interested in. But it also, activated AKT also has huge signaling effects on cell growth, proliferation, anti-apoptosis, or stopping cell death, and these are known as mitogenic effects. So knowing what's happening with the dynamics of the AKT activation, which depends not only on a chemical response, but also on where it is very important. So what we're going to look at today is where it is, because we can get some insight into that because we can see from the outside in. And with the plasma membrane being here and the activation of AKT occurring just below it, we can look at the cell membrane and just look at the AKT that is arriving there ready to be activated. To do this, we use total internal reflection fluorescence microscopy, or TIRF. And so broadly how it works is that you have a cell, and now the cell membrane is at the bottom because this is a microscopist's view. And so this is an inverted microscope where your objective is coming from the bottom, and you have a cover slip and you have the cell sitting on top of it. So you have the plasma membrane sitting at the bottom here on its cover slip, and here is where your objective is. This is the inside of the cell here. Then what we do is we think, well, there are various proteins we're interested in. This may be our AKT. And you cleverly get some people to genetically engineer this molecule, but with an additional tag on it, which fluoresces when exposed to a particular wavelength of light. And so this is then retrovirally expressed in the cell. In other words, you actually tell the cell to make these molecules, so they're in addition to their own endogenous versions of that particular protein. And then you can image them. So how do you image them? Well, you know that the cytoplasms maybe your refractive index about maybe 1.3. This is a bit fuzzy, of course. It's a very complex space. It's not uniform. And your cover glass is about 1.5. Now, let me just continue a bit too fast, I think, but never mind. What we've got is the microscopist puts in a laser light, which is incident at greater than the critical angle. And so thus the light then is reflected, totally internally reflected. But that does not mean that all of the energy is reflected. There is a certain component, and if you look at Maxwell's equations, it's actually carried in the imaginary component, the amount that's carried into the sample, and that's what's known as the evanescent wave. Now, the evanescent wave's energy drops off exponentially, and that depends, as I've written up the top, on the initial energy and also the depth into the sample. Now, the penetration depth is a function of the angle of incidence, the frequency of the light, and also the relative refractive indexes of the two. But broadly speaking, you're talking about 100 nanometers in the sorts of setups that we're seeing. Now, if you see about 100 nanometers, the energy in that evanescent wave dropping down exponentially is sufficient to cause the fluorophores to fluoresce. And so what we've got is an experimental system that can tell us when a molecule is in this TIRF zone. In other words, we're in around about 100 nanometers of the cell surface. So now we've got a measurement system where we can say, here I can observe the entry of these molecules into this area just inside the cell underneath the plasma membrane. Okay, so some colleagues, Dougal Norris and co over at Sydney University did some nice measurements. They were interested in designing different fluorophores for imaging AKT. And they did some measurements where they used this technique on multiple patches of cells, because you only get to see a small section as an individual cell when you do this. And they had a cell which was just sitting there, no insulin at all. Okay, and they let it then respond, and it then translocates AKT at a given rate. Just because you don't have any insulin in the system doesn't mean that the AKT is not moving around and being phosphorylated. There's lots of functions that it does. So there is a normal amount of AKT at that plasma membrane when there's no insulin. However, when you apply insulin to the cell, or cells in your well, and they're putting a physiological level of 1 nanomolar of insulin on here, you suddenly see a rapid increase in the amount of AKT in that TERF zone. So this is AKT being recruited to the underside of that plasma membrane of the cell. And it goes up, goes, and then comes back down. This is, remember, you were tracking the amount of fluorescence in the TERF zone at the plasma membrane. So it's overshooting, it's getting a whole lot, and then some is leaving and it's then settling down. So this is a dynamic balance of stuff coming in and stuff leaving at the same time. So this is the sort of data we can see looking from the outside in. Now, if I want to try and understand this process, because clearly being in the right place, this underside of the membrane means that this AKT can be phosphorylated, then I need to think, what sort of things do I need to invoke to describe what's going on? So if I look at this system, I can see that it has a component. It starts at a particular level, it increases rapidly, overshoots and comes back down. Now, if I'm thinking about this, this is AKT recycling around in the system. It's not being synthesized and degraded on the timescale of interest. So for my purposes, it's approximately conserved. It's just where it is that is controlling this dynamics that we're seeing. So our minimal model, and this is the work of Kathy Gray, who has just completed her PhD in the last year, and myself. So we looked at this and we thought, well, it's actually got to have at least 3 compartments. If we think about it as stages, right? We've got the AKT at the plasma membrane, right at the underside. That's what we're observing, okay? And of course, there's stuff on the inside, right on the inside, and the stuff that's just before it. Well, why? Why do I have to have this pre-primed pool? It's because I can see it's an overshoot. If I only had 2 pools, an outside and an inside, I would not be able to get this overshoot behavior. So this is the minimal model that I require to replicate the observed dynamics. So what have I done to get from basal state to an insulin stimulated state? Well, I've again tried to think what is possible to cause that change. Maybe we've got some cycling rates that take the AKT from the cytosol into this primed pool, and then from the primed pool into the TERF zone, and then back again into the cytosol. What is the minimal sort of thing that I can do that when I apply the insulin, I get this rapid rise? Well, I can change the rate at which it goes from the primed pool to the underside of the membrane. And I don't have to change anything else. Of course, it's a cycle, so I could choose which rate I might change, but the minimum thing I have to do is change something. So I alter, as a step, my rate of release, this K2 here, from this primed pool to the underside of the membrane into the TERF zone by making K2 change. And it's not a bad description of the system, but it clearly misses something at the start. It doesn't look like this system really rises back where my model is, but rather a little bit later. And why might that be the case? Is it reasonable to think, well, actually, the signals being propagated, how did they get this data? At t=0, they put 1 nanomole of insulin into the dish that held the cells. And then we saw a translocation of AKT. But it looks to me like it's and then takes a small amount of time. So we tried to look at allowing a delay. Just really, of course, just wait for a delay and then do your change in K2. And indeed, it's a much, much better fit. It not only captures that upstroke, but also much better the actual overshoot. And so that delay was about 0.4 of a minute. This could well be an extra piece of information that we've got now, because we know there's a whole cascade of events from the insulin receptor binding to the insulin, forming a complex going through IRS1 and down through PDK and down to AKT, causing it to move from the inside of the cell to the outside. So what this tells us is that that cascade of events to trigger that translocation takes about 0.4 of a minute. So now we've actually discovered something purely by considering the system from a minimal point of view. The data is relatively sparse, although the system is incredibly complex, and most of it's unobservable. But nonetheless, you can still get really interesting things that then say, okay, now I have a constraint. I actually know that all of those signaling events have to take less than 0.4 of a minute in order for this observation of the response of the AKT translocating to occur. So that was quite nice. And indeed, you could argue, well, is it something to do with the insulin having to diffuse to the cells? Is it experimental error that's giving you this 0.4? Or is it indeed the signaling delay before this system translocates? And it could be the experimental constraints and so on, but we didn't just have this data set. What the experimentalists did was actually continue on and do a second stimulus after the physiological 1 nanomolar of insulin. They applied at 10 minutes into the experiment 100 nanomolar of insulin. Now that's well beyond the value that any system would ever see, right? This is seriously overstimulated. But you can see that this is our original transition that we saw before in 1 nanomolar insulin. The system is still able to respond, and it then ramps up with more and more AKT being recruited to that underside of the plasma membrane. And so it's still possible for this system to continue to respond. How can we think about this in terms of our model? Initially, we said, okay, we can just say we're going to increase the rate at which we release the AKT from the primed pool to the plasma membrane in order to actually get that response that we saw with the 1 nanomolar insulin. Is it possible that simply ramping up that release rate more and more and more might indeed show us that response when we have a much, much higher level of insulin? Now that we're in the 1 nanomolar state, can ramping up K2 more give us the response we're seeing at 100 nanomolar? So we do the test where we fit our initial response, and this is just because we've got a step function in K2. It's at a lower level in the basal state, no insulin, and at 0.4 of a minute, we actually allow it to step up. And this is the fit that we found that really described the data well. If we now, at 10 minutes, allow K2 to again step up in value, this is the sort of thing we're going to get. So all other parameters are remaining the same. So increasing K2 again at 10 minutes causes another rise. However, keeping on increasing it, because it's clearly not at the same height, is not going to cut it. And so it's a bit disappointing in a way. Our hypothesis was maybe you see this might work, because in order to then have this slow rise, which we're really not seeing here, maybe there might be some point at which this pool, X2, is drained, in which case then the dominant rate becomes this K1, and that then allows us to feed in, and we swap from K2 controlling the timing in this second stage to K1 controlling the timing. So this is like a depletion hypothesis. And so we tried relaxing all of the constraints on the K2 and then trying to see if we can just increase it, and trying different combinations of K1, K2, and K3 to see if we can get this to fit. But no matter what we tried, our K1 was always in a relation to K2 that did not allow this to happen, because we've got K1 always being less than K2 in all of the scenarios. And so it was not possible to get this slow rise that we're seeing at 100 nanomolar from just having K2 being the change and then having it depleting our pool in the primed pool and then having a delay. So that was very disappointing because that was my hypothesis. And I thought, oh, this is a really good explanation, but it wasn't to be. And as a modeler, you've got to roll with those punches. And then you've got to say, well, what else could it be? So our next assumption was to say, well, we know that changing K2 when you apply physiological insulin allows us to model that physiological step. So our K2 value is fixed to a certain value in the basal state, and then it steps up when we take and apply insulin to the system. So we're going to leave K2 as it is, right? So this is a retention mechanism we're trying to model. What we want is a slower rise in the amount of the plasma membrane compartment of AKT when we put in 100 nanomolar. And so we can't have it disappearing as rapidly as we saw when we just stepped up K2 again. So let's just step it up because we know that that works for the 1 nanomolar, but switch now our attention to K3. And this is the return pathway. So if we allow this return to change at high insulin levels, then we can change the balance of the inflow and the outflow, because the amount here is going to be the balance of what comes in and what goes out. So our retention hypothesis allows K2, the release from the prime pool, to be instigated by insulin. And then when you apply further high levels of insulin, it allows the return pathway to be altered so that we retain more at the plasma membrane. In other words, if you reduce K3, then you end up retaining more at the plasma membrane. So, of course, there are other combinations. This is a cycle. So you could do different changes in these particular rate constants for mathematically exactly the same effect. But what we're trying to replicate is an initial overshoot in the plasma membrane level that settles down when you apply 1 nanomolar of insulin. And then when you furthermore apply 100 nanomolar insulin, that you get a slow rise, which actually for the data set that we've got is continuing to rise. It looks, it doesn't look like it's an overshoot. It looks like it's just a smooth rise to a final state, which we can't observe with the data set. Okay, so here we go. Here's the data, and our hypothesis says, well, we're going to have K2 changing to describe this bit, and then we're going to leave K2 in its insulin state. So it's just going to have two values, either 0 or insulin, and it doesn't care how much insulin. And then we're going to have a further rise. Now, this is our first hypothesis. What if that delay is due to the experiment? Okay, so the 0.4 minutes is because you're dropping insulin on, it takes a while for things to change, and then it's got to go down into the cell. Well, the 0.4 minutes absolutely fits that first rise beautifully. That's where we got it from. But here you can see, if you say, well, it's going to take about that long for the insulin to get into the system and actually act because of the experiment, then actually it's delaying it here after the 10 minutes. And it's not a bad fit, but it doesn't seem to get that upstroke again. So then we want to pursue perhaps saying just any old delay. Let's look at a free delay. Okay. And it turns out that that free delay is very, very small. So being an Occam's razor advocate, well, why have any delay at all? Here's no delay on that second stimulation. And you can see that the fit is remarkably good. So let's just compare all three together. The outcome that we think that we have here is that we have got some handle on the delay of the signaling system as any amount of insulin primes the system to actually respond. So it activates the signaling pathway and activates translocation. This is encoded by a change in this rate from the primed pool to the plasma membrane. Subsequent applications of insulin, higher levels in this case, don't require that priming step because the system is already activated. Remember, low levels of AKT activation also have downstream ramifications. So we only enact the translocation system in that second stimulation because it's already ready, primed to go. And indeed, you can see that the data is actually still on a rise. And so the no delay is also able to capture that a little bit better. Naturally, and this is what every modeler says, I'd love more data, right? I want to know what happens next, okay? But of course, these systems are real and have limitations. A, you can't run things forever. B, this is not actually the purpose this experiment was done for, but also, unfortunately, those beautiful fluorophores also bleach over time. So you actually, the more you image, the less the thing glows. And so therefore your bleaching effects are going to get more and more. And so you then have to adjust more and more to account for bleaching effects. And so the data doesn't become as robust down the track. So what we have found though, is that it is possible to think about this, to say the full time course of this data is if the effective recycling rate, the K3, is decreased following the application of high levels of insulin, we are going to see this system being well described. So that there are clearly two stages we've got: a signaling event, which triggers translocation, and then we see a slower, and it's reasonable that it's slower, movement of actual molecules from one location to another. So it's pretty clear that biochemistry is fast, but movement is slow. So presumably then the signaling delay is shorter than the translocation delay, and thus we've got this. So we've got then this system working a little bit like a dimmer switch, right? Any application of insulin is the power on and off, and then the level of insulin ramps the dimmer up and down that controls the final level of the actual AKT at the plasma membrane. Now, ultimately, we're not looking at whether or not this AKT is phosphorylated or not, but knowing that it can only get phosphorylated at the plasma membrane, then knowing that it's in the right place means that we can then look at what might happen in terms of phosphorylation and put this translocation model into a system which then takes the phosphorylation and then downstream signaling into account. So this is a paper that Kathy finished her thesis up with, and it's appeared in the Journal of Theoretical Biology, if you're interested. So I got to thinking with this, of course, that timing is really useful to know the order of events. And so I then thought, well, this would be interesting to see whether or not any components that we track that we know are downstream of AKT also have this sort of timing delay before they react to an applied signal. So my molecule of interest for a very long time has been GLUT4, which is glucose transporter 4. It is the transporter that's responsible for glucose uptake in muscle and fat. It's driven by an insulin signal, And AKT is definitely a key component, low threshold switch that lies in its signaling pathway. So what do I know about it? Well, after many years investigating, and here's a nice little picture of what GLUT4 looks like, but we found that the GLUT4 action is correlated in terms of its dose response with not AKT in general, but AKT at the plasma membrane. Okay, so this translocated version of AKT that we've just been looking at is the driver of downstream effects for GLUT4. I also know that GLUT4 has a relatively complicated cycle. So if we're on the outside of the cell, in order to get GLUT4 on the outside of the cell, we have to translocate it in little membrane bubbles up to the underside of the membrane, whereupon those membrane vesicles fuse and join into the membrane, expressing GLUT4 at the surface. And it recycles around. So there's quite a few different steps that need to be in there to understand the system. But we've got some handle on it. So here's our GLUT4. It's membrane embedded. It doesn't live outside of the membrane. It doesn't like it. It's got hydrophobic regions, which then get embedded inside the lipid bilayer. And of course, people have added lovely tags to it to again glow in the dark. All right, so the nice thing is, and again, this is a microscopist's view, so the cytosol or the inside of the cells up the top and the outside of the cells down the bottom. If you add this nice tag, you can also add secondary conjugations so that you can use double marking on this type of molecule. You can express it in the cell and then use antibodies to activate the fluorescent markers. So here we've got our cell, and I've gone back to the usual view where the outside of the cell is up the top and the inside of the cell is down the bottom, and here's our plasma membrane. And so, oh gosh, that didn't work at all. Oh dear. My animation's not working, so I'm not going to bother. Basically what happens is you've got molecules in vesicles that come up to the surface. As they hit the surface, you're going to then be able to visualize them, not necessarily using TERF. In this case, we're using flow cytometry to actually then count the number of molecules that are on the surface of the cell as they arrive in response to insulin. So as a function of time. And so this is data from my collaborator, Cynthia Mastic, over in Reno. And she's measured as a function of time, the surface level of the GLUT4 molecule. And so it's noisy, as is all biological data. And so what happens is that you get, for 0.3 nanomolar, so on the low side of physiological levels of insulin being applied at time 0, you get a rise in GLUT4 levels on the surface. And that would be in response to the insulin signal. This is what you'd want if there was glucose in your system. If you add more insulin, so this is for 3 nanomolar, you get more GLUT4 on the surface. And you keep on increasing it, you get more on the surface and more, although 30 nanomolar and 100 nanomolar are pretty much the same thing in terms of the long time response, because they're well over any maximum level you'd see in physiology. Are we going to see any delays here? And this is something that I'm just playing with at the moment. So let's go back to the 3 nanomolar cycle. So it's going to cycle GLUT4 out in response to the insulin being applied. So broadly speaking, although it's somewhat more complicated, you could look at it as a exocytosis or outward flow process and put a nice rise into it with a compartmental cycle. Now, that looks reasonable. It's very noisy, but so too is a delayed version of that. And, you know, you might not be convinced that why should you increase the complexity of your model by putting a delay in for this? Well, let's zoom in a little bit and have a look at the early data. And it's somewhat more justifiable when you look at this. you're increasing your complexity, allowing one more degree of freedom, but you may then gain something. I'm not saying that this is robust, this is a preliminary study, but it does seem reasonable that we could see a delay. And so this delay turns out to be about 0.9 of a minute. Remember, GLUT4 is downstream of AKT, and this is 0.3 nanomolar of insulin being applied. So 0.3 nanomolar of insulin on, measure the response of GLUT4 right down the end of the cascade in response to that. Okay, so this is a similar measure, but for 3 nanomolar of insulin. So a higher level of insulin. And I think you can probably see that it's tighter at the start. And so this delay appears to be more justified. Now, remember that these are measurements from very many cells. Okay, so we're seeing a mean field response here. There may be high and low responders, fast and slow responders in these cells. They're well known to actually have these different insulin-based effects, to be heterogeneous. So nonetheless, I think it seems reasonable that we could be seeing a delay here. And it's particularly in focus when you go to higher and indeed higher levels of insulin, especially at this maximum level of insulin, unlike in the AKT where the 100 nanomolar extra step didn't do anything at all. This is a step from 0 to 100 nanomolar. And so we're seeing a delay for things to happen. So if you go straight from basal to an insulin level, our AKT model says you need to get that signaling happening and then translocation happening. That took about 0.4 minutes for our physiological level of insulin. And then our regulation kicked in after that. So what's interesting is for these different insulin levels, the delay for the GLUT4 response, even though it's a bit coarse and it's just a preliminary look, appears to increase with increasing insulin. So let's put that in context with the AKT that we saw though. The AKT delay was about 0.4 of a minute, and that was at 1 nanomolar. And so, nicely, that delay is less than the delays we are seeing on a downstream effector. So perhaps what we've got is a divisor. We've got about 0.4 of a minute for the upstream effects, upstream of AKT from the outside of the cell in, and then the remainder that we're seeing between the 0.4 and these levels is the downstream signaling propagating down from AKT being activated through a whole cascade of events which are really not clearly understood to actually cause the translocation downstream of GLUT4. So we've now got a handle on the sort of timing that those biochemical events in the signaling pathway may actually be constrained by. So that gives us a real opportunity to now divide up that network and look at the key players and look at how that signaling might be affected. We've got data about what are the key things that affect, for instance, GLUT4 translocation. So a component AS160, for instance, is a known point of change at key stages of the translocation cycle. Now we can have the opportunity to look at the timing of its activation relative to the activation of AKT and put some more points for that delay system within this network. So this opens up the idea to map the delays of the known components to try and then get the order in which they operate and perhaps then also where they might be operating, because it's not just the timing, it's the location that's important as well. And indeed, it's very much an ordered system because multiple components may well need to be at the same place at the same time in order that a downstream effect can be enacted. There are also clearly stochastic effects here. And one of the big things that I'm working on is the development of techniques to look at how one might measure the difference between stochastic data and then a stochastic model, because mean field models are terrific for getting an overview, but the system is itself very heterogeneous. So in the end, when you have a handle of what's going on from a mean field perspective, it then behoves you to actually look at how those things might be being enacted. And for that, you really need to take a stochastic perspective. But once you have a stochastic model, you need to have techniques to compare your stochastic model with your stochastic data in a robust way that can drive parameter inference and indeed model selection, because that's what it's all about. Why would you take one model versus another? What gain do you get from it? I'm very much a reductionist. I take the simplest model and build it up until it explains, but in doing so, you then know what the primary determinants may be in your possible version of the truth, which of course is what any model is. So with that, I'd like to thank my collaborators, Cynthia Mastic in the University of Nevada, who's done the Group 4 data, and of course, Kathy Gray, who's now finished her PhD, and I miss her a lot. All right, thank you very much.